we didn't do the homework for the
Yeah.
package. Yeah.
Yeah, I don't know, do it at your own pace. Okay, today we're going to be learning about biotechnology. Uh, DNA tools basically just means like the way we manipulate DNA. So what is biotechnology? It is the manipulation of biological organisms, and usually we do that by manipulating DNA itself. So if you look here, there's different branches of biotechnology. Okay, so obviously there are techniques to manipulating DNA, and we are going to get this far today. So we're going to do all this stuff, and then this stuff we'll do tomorrow. So I'm pretty sure you've gone through uh, isolating DNA before. So I think last year in bio or chem, you may have done an experiment where you get, I think, banana, and then you isolate
Yep.
the DNA from it, if I remember correctly. I remember doing this. This is one way to isolate DNA. So what is the point of isolating DNA? Before you can manipulate DNA, it actually has to be isolated from the cells. So there are many steps, there are a couple steps you have to do to isolate DNA. Step one, you have to disrupt the membrane, the cell membrane, with some kind of detergent. Then you filter to remove the larger pieces. Then you can precipitate DNA with ethanol and then retrieve the precipitated DNA. So, while in school or in grade 11 biology, you may have done these steps as listed, like detergent, then you filter to remove the debris, and then you obtain the DNA through ethanol precipitation. At the science center, you would have done it a little bit differently. So, at the science center, what you would have done is you would have used heat to disrupt the membrane, and then you would have centrifuged to remove or to separate the DNA from heavier cell parts or larger cell parts. And then in terms of actually obtaining the DNA, you would have kept it in solution and then visualized your DNA on a gel.
But at the science center, where are we getting the DNA from?
So what you actually do is you swab your cheek, or you did a couple options, but I think everybody ends up swabbing their cheeks. Like you talk about the different options, but swabbing your cheeks is the best option
I see.
at that point because it's easy to obtain, it's not invasive. Um, so visualizing on a gel, at the end of this PowerPoint, we're going to learn about something called gel electrophoresis. We will learn about that tomorrow, and it's a way to, actually we'll just learn about it, but visualizing means using gel electrophoresis to see the DNA. All right, so right now we're going to talk about DNA amplification. So after you've isolated your DNA, let's say you have DNA from a couple cells from your cheek. It's much different from using like part of a banana where you'll have like many cells. Like in your cheek, you can get a couple cells, but you can't get like a whole lot of DNA, right? Some processes like testing or like even visualizing on a gel, you need more DNA than you can obtain from just a cheek swab. So what do? You have to amplify this DNA. PCR, or polymerase chain reaction, is a way of doing this in vitro. So not in a living organism, or literally like in glass or on a petri dish, essentially. I think when we refer to in vitro, it just means like out of a cell. So the purpose of this amplification is to increase the amount of DNA we have, or create millions of copies of a specific DNA sequence synthetically. It's essentially DNA replication in a tube. So, what are the major steps in DNA replication, and what substance would you need in each step of DNA replication? Because, as I said earlier, we're not doing this in a cell, so we need to identify the major steps and what we'll need to do this outside of a cell. I want to ask you guys to brainstorm here. Major steps: unwinding, priming, elongation, and termination. Naturally, all of this occurs because we have the biological machinery, but if we wanted to replicate this outside of the cell, we use a technique called PCR. So, let's go through this. Normally, you start replication at an ORI on the DNA template, helicase comes on and unwinds the DNA. In real life, we provide PCR, the DNA template, and we use heat to unwind the DNA. For priming, we use RNA primers with a primase, but in PCR, we use two DNA primers and something called an annealing temperature. So we set the solution to a temperature where our DNA primers will actually just anneal to our DNA template. Elongation is done by nucleotides and something called an enzyme called DNAP. PCR also uses nucleotides and something called TAC. And then termination, when it reaches the end, meets another replication bubble. But in PCR, we know to terminate by just changing the temperature again.
Wait,
So
what does PCR stand for?
PCR means polymerase chain reaction.
Oh.
Yep, it's fine. So we'll go through more closely. So what does PCR need? Overall, we need a DNA template. So this is the target strand on DNA that we are going to, or the target sequence on the DNA that we want to amplify. We're interested in amplifying. Then we'll need nucleotides. Then we'll need a DNAP, but in this case we'll use a specific polymerase called TAC, and we are going to need DNA primers. So the PCR cycle, PCR is done in cycles. A single cycle of PCR involves step 1 denaturation, step 2 primer annealing, and step 3 synthesis. You're going to need to know these temperatures. The temperatures are essential to the steps themselves, so you're going to have to know. Each source where you use lookup PCR might give you a different temperature, but for our purposes, you can just learn these numbers because generally they're correct. Do we go over? Okay. So I guess we don't go over it in separate slides. So let's refer to the diagram on the right with the steps here. What is denaturation? So denaturation is you briefly heat the DNA at 95 degrees Celsius to interrupt the hydrogen bonds and separate the DNA strands. And then in step 2, you go to a different temperature where you want to achieve primer annealing. So the DNA primers that we referred to earlier that we need, these guys are like floating around in the solution with the DNA. So once the DNA strands are separated, at this temperature, will allow the primers to form hydrogen bonds with the ends of your target sequence. And they bond because we have specific primers because we know the target sequence, so we know what our primers need to be to attach to the ends of these target sequences. Step 3, synthesis. So in step 3, our DNA polymerase, or in this case TAC polymerase, will add nucleotides to the 3' end of each primer, and that is one step of PCR complete. So if you look here, after one step of PCR, you go from this target sequence to these two, and then after the next step, you have 4 molecules, and then step 3, you have 8 molecules. But in these white boxes, we call these, the white ones are important because the white ones are what we want. And I'll talk about this a little bit more in a second. So essentially, when you have PCR, or when you do PCR, you have the entirety of the genome. So let's look over here. You start with basically the entirety of the genome, and you have a target sequence that you want to amplify. You only want to amplify this target sequence. This is what you're interested in for PCR. It's only at step 3 that you have molecules that are the correct size, that match this target sequence without all of the other stuff. So what does that actually mean? So let's assume that we start with just the entire genome, and this is the target sequence that we want. After step 1 of PCR, this is the original with the black and the purple here. The orange is the stuff that we've just created. If we look at this entire molecule, is this matching exactly the target sequence that we want? No, the answer is no, because on this strand, the bottom strand here, we have this extra information that we don't want. Even in step 2, we're a little bit closer. In step 2, we have molecules that almost look like the target sequence that we want, but we still have this extra information that we don't want, and it's only in step 3 that we have exactly the sequence that we want. and that's and that's because of the way that PCR works. Let's go to Paint. All right, let me open up Paint. All right. So, at the Science Center, you guys would have done an activity that goes through PCR step by step with essentially materials that would help you see DNA and what's happening to DNA in real time. But since you guys aren't at the Science Center, we're gonna have to do this in Paint. So try and follow along. We'll go through PCR as I try my best to draw it out. So what materials do we have for PCR? We have the original template. So I don't know, we'll say template. We have, nucleotides. So we'll just call them DNTPs. Then we have TAC and we have primers. And remember that the primers are DNA primers. We do not have an enzyme to put these down. We anneal them by temperature. And then we have something called TAC. Cool. So let's start with our entire genome. So this is the template. And we're going to pretend that our template extends in both directions. But I won't keep these arrows here because it get a little bit confusing. On this template, we have a specific sequence that we are interested in. Let's say that in here is the gene that you want to test to see whether or not you have like sickle cell anemia. But in order to see if you have it, you have to amplify it. Let's just say hypothetical situation. Let's go through step 1. Step 1, the strands separate. So I'm going to put arrows like this to show that they continue, just to differentiate. Step 1, they separate. And here I'm putting these lines to basically tell you where the target sequence is. And how about I add some 5 prime, 3 prime. Okay, this is denaturation. And after this, we're going to do annealing. But I'll just draw the annealing on here instead of drawing everything again. So when we anneal, we have our primers, and they attach at the ends. But the primers won't attach twice to the same strand. When we say the end of our target sequence, remember the job of the primers is to give something for polymerase to build off of. So if we think about it that way, our primers will attach closer to the 5' end of this target sequence. Because if it attached here, for example, this end would be 5', and DNA polymerase would not be able to use this primer to build the rest of the target sequence that we want, right? It wouldn't go this way. It's important that the primer binds to this side of our target sequence so that polymerase can extend and recreate the target sequence in the correct direction. So this is what happens, okay? Our primer is anneal. And then in this case, TAC polymerase comes along and recreates our sequence. But let's say that TAC polymerase comes and recreates our sequence, okay? So what I'm going to draw from here is I'm going to draw this strand and this strand. So let's draw them. It's continuing in this direction because polymerase would have continued to the end. Wait, did I do this properly? Give me a second, give me a second. I don't think I drew this properly.
Does DNAP build from 5 prime to 3 prime?
See? I I knew it. Okay. There we go. My
Yeah,
explanation.
you were doing it on the template strand, but the nascent strand goes the other way,
Exactly.
I think.
No, you're right, David. I drew it incorrectly. There we go. And then it would continue this way. Right? I think I got it right this time. Yeah, sorry about that. That must have been pretty confusing. Here, let's draw it this way. This is 5 prime. This is 3 prime. Good, because, okay, anti-parallel. The DNA template is 5 prime to 3 prime in this direction, so the complementary should be opposite. Sorry if I confused you guys. Good thing you caught that. Okay, so I'm going to draw them. The complementary strands that we just created are the ones that I'm drawing. So the complementary strands that we just created has an end here, so it's different from the originals that we started with. So the ones that we just created has this end. This is the 5 prime end, but the target sequence still ends here. And then for the other one, it'll continue like this, has its 5 prime end here, and the target sequence will still end here. Oh, maybe I should draw it like this, or like this. So in both of these strands, we have one end of the target sequence the way we want it, but we still want to cut this part off, or we don't want this stuff. So we need to go through another step or another cycle of PCR, because what will happen at the next step of PCR? So over here, the primer will attach, and this will be the 5 prime end on the primer. And over here, same thing, 5 prime end of the primer. And then Taq polymerase can come and continue it to here, and then continue it to here. And then by the end of our second step of PCR, we'll have, wait, no, yes, by the end of our second step of PCR, we'll have 1/2 of a correct DNA molecule. And after our third step, after you use this as a template, will you have correctly sized DNA molecules? Does that make sense to everybody? So that's why over here we say that cycle 3 yields 2 molecules that match the target sequence. You will have many other strands, and in my paint example I only focused on creating these 2 white ones, but this is what we want essentially. Does it make sense to everyone? Okay. And
Don't you already have, sorry, don't you already have strands that are the right size by cycle 2, not cycle 3?
so cycle 2 you have half. So you do have strands, but you don't have a whole DNA molecule. So you have one side that's the right size. Does that make sense?
Oh, okay.
But you want an
Okay.
entire DNA molecule that's the
Sure,
right
sure,
size.
sure. Sure.
Does that make sense to everyone? And all these other ones, like all these other ones will eventually end up creating the matching sequences. It's just that at cycle 3, we have 2 that are the correct length. So this is important to understand. At which cycle do you get the correctly sized target sequence? So this is just another. Okay, so let's make some comparisons here. So DNA replication in the cell or in vivo uses RNA primers. PCR uses DNA primers. So why is there this difference? Why can PCR afford to use DNA primers, but DNA replication can't? Do you guys have any answers? Anyone? Any guesses?
No. Sorry. Not all of us are that smart.
Oh, it's fine. So let's go back to step 2 of PCR. Step 2 of PCR, primary annealing. In step 2, we allow temperature to dictate when our primer is attached to DNA. Because when we first denature DNA, we heat the solution to 95 degrees Celsius. And then to allow our primers to hydrogen bond to the target sequence, we cool the solution to 50 degrees Celsius to 65 degrees Celsius. There are no enzymes involved here. Does that make sense to everyone? We have specifically prepared these DNA primers to match our target sequence where we want them to bind to our target sequence. This is the key difference from DNA replication in the cell to PCR. We have specifically curated these primers per se. There are no enzymes involved. If we used RNA primers in PCR, we would have to basically add extra materials or extra steps to this replication cycle that are unnecessary. Because if we used RNA primers, sure, they could also anneal to our target sequence, but we'd have to add another enzyme that would have to come along and replace these primers. So the entire process would be more complicated.
So why don't cells just use DNA primers? Why do they need to use RNA primers?
Because my best guess would be that they can't, most cells can't afford to denature at 95 degrees Celsius, then anneal at 50 degrees to 65 degrees Celsius. Like, we have the ability to carefully control the temperature and the situations, but cells don't, so they use enzymes to do this instead. But I can't tell you why they use RNAP and RNA primers over just using another DNA enzyme that could add DNA primers automatically. Oh, exactly. Okay, that makes sense. DNA primers, Nathan just said that DNA primers can't start on their own strand, but RNA primase and RNA primers can. Yes, yes, Nathan. Thank you. You're very smart. Okay. So if you look over here, these DNA primers have also started by themselves. And remember the key difference between DNAP and RNAP that we learned in DNA replication? RNAP, so if we have a template like this, RNAP can come along and put down RNA nucleotides without an existing 3' end to add to, right? But remember that DNAP can only add nucleotides. Oh, I mean, this is confusing. So, like, this 3' basically means that there's a 3' end of another nucleotide here for it to work to, but RNAP doesn't need this. But DNAP does. Remember, guys, that when I explained in DNA replication, DNAP uses these RNA primers because DNAP can only add to an existing 3' end. But in this case, oh, it looks like a 2, but in this case, there is no like end to add to. And in the body, there would be no 3' end for a DNA primer to be added to, if that makes sense.
So is it that DNA primers need more energy to be added, and here we're getting that extra energy because there's more temperature?
I don't know if it's about energy or if it's just the way that the enzymes are.
But how come DNA primers can be added without, like, being added? Like, how come they can be added directly without
Oh,
RNA
okay, now
primers?
I understand. It looks maybe that is true. So maybe they do need a higher heat because DNA primers can anneal by themselves in this situation, but in the body they wouldn't be able to. Or maybe this reduces error. In PCR is prone to error. And maybe using the RNA primase and putting down RNA primers and then DNA primers and replacing it leads to less errors. Because that could also be a very genuine reason to do it that way. Because errors in replication is a big deal. If you have many errors, then you could have a inviable like organism, right? So yeah, I can
How
find
would
that.
this how would this create more errors? Would it be because primers are attaching where they're not supposed to be?
Yeah, primers can attach where they're not supposed to be. So also here we've carefully created these primers so we know the sequence on the template. So we know what our primers have to be, if that makes sense. So obviously the body has ORIs, and those ORIs have specific sequences to begin replication, and then the primers come along. But I don't think that the body would be able to create like a specific primer for a specific spot on like basically billions of places on DNA, if that makes sense. This is a more carefully controlled environment.
All right.
Does that answer your question?
Um, I don't exactly remember what my question was now.
Okay. Uh, all right. So I guess that was pretty long and complicated, so let's just overview. Why can PCR use DNA primers? PCR can use DNA primers because A, it uses temperature to anneal them and not a DNA polymerase. And B, DNA replication can't use DNA primers because of the properties of DNA polymerase, essentially, because RNAP can just put down a nucleotide wherever it wants, but DNA nucleotides can't be put down wherever. They need an existing 3' end that isn't there. Does that make sense? This is a little bit confusing, but I'm sure the more that you guys look over it, it should make more sense. Okay, Nathan, do you have another question? So Nathan is saying, so it can get past that limitation, and the limitation he's referring to is the DNA polymerase that needs a 3' end. It can get past that limitation because no enzymes are involved, and yes, that's exactly right. It can get past that limitation of DNA polymerase because it's not here. We don't use DNA polymerase. We use heat to anneal the primers. Yeah. Alright, so DNA replication in the cell terminates when you get to the end of a chromosome or meets another replication fork. How does replication terminate in PCR? And what stops the DNA polymerase in PCR? Do you guys have any ideas? How does replication terminate?
I think it's just the end of the chromosome. And then
Okay,
when the DNA is replicated, the end becomes much shorter.
so let's look over here. So in this case, so let's look at cycle 2. At cycle 2, specifically, um, let's move this out of the way. Let's move this out of the way. So specifically, this molecule here, let's say we're replicating the right side strand and DNA polymerase is moving in this direction. Yes, it reaches the end of the chromosome, per se. It reaches the end of its template, so it has no choice but to stop. But what about cycle 1? So cycle 1, if we look at this molecule, Taq polymerase or DNA polymerase is continuing in this direction where it should have space to go for a very long time. Like we're not going to leave it to go all the way to the end of the genome because we don't need that information. So what we actually also do is we control the polymerase, we control the enzyme using temperature. So I don't know exactly how they'll know when to change the temperature, but synthesis has a different temperature from denaturation. So when you want synthesis to end, you can increase the temperature and the enzyme will stop. Does that make sense, guys?
Yep.
So that's another way that they could control it using temperature.
Wait,
So
but if you increase the temperature, doesn't it just cause it to break up again?
That's
so
what you want.
yeah, you do want it to keep breaking apart because you want to keep creating new molecules.
But what about at like the end where you don't want to create any more new molecules?
Um, you basically want to create as many copies as you can. Um, is your question like, so let's say synthesis is running and we run another cycle and we denature this DNA again. Aren't we destroying the DNA? No, we're not destroying the DNA. What we're doing is, let's say at the end of cycle 1, which yields 2 molecules,
Hmm.
after synthesis of, cycle 1, we're going to go into denaturation of cycle 2. So what's going to happen? It's going to split like this, and 1, 2, 3, 4 become the templates for 1, 2, 3, 4.
Yeah.
So we want this denaturation. Are you referring
All
to
right.
the DNA or were you referring to the enzyme, DNAP?
Um, I was referring to how like let's say you wanted it to stop at cycle 3.
Mm-hmm.
Um, if you increase the temperature, wouldn't that just cause it to go to cycle 4?
Oh, okay. So you have the control to end it at like a certain cycle, of course. Um, yeah, actually you make a good point. For the very last cycle, my best answer would just be that many molecules would have DNAP end because the actual strand ends. And for those that don't, and those that still have like this extra information and DNAP just chugs along, or DNAP just chugs along, creating more information than is required, that's not too much of an issue.
Also, if you were to decrease the temperature, does that stop it? Like
Um,
let's say you decrease to like room temperature, let's say.
I think it might. Let's go over it because we'll go over Taq polymerase, I think, in one of the next slides.
Okay, okay.
Okay. Oh, it does go into. Okay, I mean, let's go over this again. Primers, they're a synthetic sequence, so we create them. They're single-stranded DNA, 20 to 30 nucleotides, and even here they provide the 3' end for polymerization. 2 primers are used for the complementary to each end of the target region of the DNA. And as I said before, each molecule will take 2 primers. And these primers define the length of the target DNA to be replicated. And we went through that in Paint. So, oh wow, it's here. I should have just kept scrolling. This is a much better diagram than the one I drew. Let's look at here. Original target double-stranded DNA. Primer 1 and primer 2 attached to each strand after it's been denatured. Then these primers are extended and the same process repeats until you have the DNA strands that are in the desired length, and so forth. Wow, this is literally what I drew in Paint, but it's much better. Okay, so before I was using TAC and DNAP interchangeably. So TAC is a specific DNA polymerase that is from a specific organism. So why can't we use the DNAP that is found in most bacteria? Or even human DNAP? Why can't we use it? And the reason why is because of temperature. So Taq polymerase is actually isolated from a bacteria called Thermus aquaticus that lives in hot springs. So their polymerase is heat stable and it can withstand extreme temperatures. So remember how step 1 was 95 degrees Celsius? Uh, and step 2 was 50 to 65 degrees Celsius, and step 3 was 72 degrees Celsius. So what we actually do is we put all the components into like a little tube thingy, and then they go into a big machine, a PCR cycler, and it just changes the temperature. So all the components go through these temperature changes together. If you had human DNAP and you exposed it to a 95 degrees Celsius environment, that enzyme will no longer be functional, right? So what do you do? So that's why we use Taq polymerase that can withstand this heat and we can reuse it cycle after cycle. So what did we do before we knew about Taq? We had 3 water baths at 3 different temperatures, and we would have to re-add DNAP at the beginning of each synthesis step. Why? Because first you put the DNA molecules into here. Let's say it's 95 degrees Celsius. We move it here, 50 to 65 degrees Celsius. This is where we add our primers. And then over here, we would add in our DNAPs, right? But we can't just like isolate the molecules of DNA in this water bath. Well, we just move all the water back here. But then after 95 degrees Celsius, again, all the DNAP that we used in the first cycle is now not going to work after our second 95 degrees Celsius step. So it's a pain in the butt, right? So today we use something like this, a machine like this, where you put all the materials that you need into a little like, I don't know what to call these, like plastic containers. And then we put these containers into here and then they're thermal cycled. So yeah, what it does is it has a plate that heats and cools, and the process takes about 1 to 2 hours. And after 30 cycles, we have over a billion copies of DNA produced. Do you guys understand why Taq is so important? Any questions about that? All right, cool.
Just is it because are we using TAC because it can operate at a higher temperature and that makes it faster than DNAP?
I think instead of faster, it's more like Taq has the ability to survive the 95 degrees Celsius step.
Oh, all right, all right.
And we put it at 72 degrees because that's where it's supposed to operate, I believe.
All right.
Yeah. Okay. So PCR was developed in 1986 by a man named Kary Mullis, and he received the Nobel Prize in 1993. All right, so now you've just learned of in vitro. You already learned in vivo DNA replication. So we can brainstorm some pros and cons of each method. So the in vitro method of DNA replication, or PCR, is not always the best way. Because right now, you guys have just basically learned the pros of, in vitro DNA replication. Look, 1 to 2 hours gives you over a billion copies, right? So you're like, wow, this is great, right? Like, why don't we do this for everything? Like, why even have in vivo DNA replication? Well, there's a couple reasons. So, sometimes it is better to make more copies of the DNA inside a fast replicating organism, like a bacterial cell. So let's brainstorm some limitations that PCR faces. Okay, so I guess we will cover in vivo DNA amplification first. So what do? Bacterial cells are transformed, and remember we learned what transformation is, and then they're grown, and that's in vivo DNA amplification. Okay, so it looks like we're gonna go over transformation before we compare and contrast them. So let's go over transformation. So what is transformation? Transformation is the alteration of a bacterial cell's genotype by the uptake of foreign and often naked DNA. What is naked DNA? Naked DNA is DNA not inside a cell, and thus the donor cell can be dead or non-existent. So dead, remember Griffith's experiment with I think it was Griffith. So the heat killed S or R. Yeah, actually, we'll move on. Okay, here we go. Griffith's experiment. Wow, Miss Law basically read my mind. When two different strains of bacteria are mixed together, eventually they will show characteristics of both strains. So, Griffith's experiment. He killed S with a live type R. What happened was type R went through a transformation with the naked DNA of the heat-killed S. Alright, so methods of transformation. How does transformation happen? Sometimes it's natural. So some bacteria have surface proteins that recognize and transport DNA from closely related species. This is a little diagram that shows you. This is your normal bacterial chromosome. This is the transforming DNA. Sometimes you have these DNA binding proteins, and then they come in, they take in the DNA, single-stranded DNA. So first they bind the double-stranded DNA, then they uptake a single strand of the DNA, then they recombine, and then after that the cell is transformed. There's also an artificial way to transform bacteria or transform cells. So transformations can also occur when bacteria is subjected to treatments that make the cell wall permeable. So there are a couple methods. There's a chemical method. So cold calcium chloride treatment followed by heat shocking can create holes in the cell wall that make it permeable to bacteria, or something called electroporation, where cells are shocked with an electric current to create holes in the bacterial membrane. Both of these methods just create holes for DNA to basically pass into the cell and be recombined. So I don't know, let's look over. Basically, the definition of in vitro is um, like artificial, writing it sideways, and in vivo is in a cell. We have 10 minutes, so let's cover this little slide pretty quickly. So, DNA amplification in vitro, what is it? It's PCR. And in vivo, it's, normally most of the time we use bacteria. Doesn't mean it has to be bacteria, but bacteria are fast replicating and they're alive. So, I mean, we could technically use humans, but the ethics of that sound pretty insane.
Just a question.
Yeah.
In bacteria, if you were to like introduce human DNA, how would you make sure that when it's in the bacteria genome, it doesn't code for anything?
Oh, that's a very good question. And actually, you will learn about gene cloning in one of the next PowerPoints. So, um, write that question down, save it, and ask again when we go to gene cloning because you will learn there.
All right. All right.
So, let's look at. Oh, wow, she already filled it in for us. Let's look at some advantages. So, for PCR, the DNA can be in a partially degraded state and still be amplified. But the major advantage of in vivo DNA amplification is proofreading. So there are very few errors in replication because we learned about the error correcting methods that all biological organisms basically do to their own DNA to reduce the number of errors, but PCR doesn't do this. So this is a limitation to the number of times the PCR cycle can be repeated. After, did we say 8 cycles, you have a billion copies? You don't really know how many of those copies might have errors in them. So when you need your DNA to have very few errors, in vivo is superior. But if your DNA is partially degraded, so let's say like from a crime scene, you have partially degraded DNA, right? Or your errors, the errors don't matter too much to you, then PCR is better to use. Ask away, Nathan. So long question. Lots of typing. The suspense. So it says here that, oh, Nathan is asking that it says the primer defines the length of the target DNA to be replicated. So does that mean it's another way to control the termination, or am I misinterpreting that? Okay, so I think you might be making a small misinterpretation. I can tell you exactly what it means by the primer defines the length of the target sequence. So it comes from the idea that you have two primers, so they're not identical. You have primer 1 and primer 2. So the way that the primer defines the length of the target DNA is that when you have your original double-stranded DNA and then it separates, in strand 1 a specific primer will bind, and then in strand 2, so the other strand, another different primer will bind. And these two primers together define the total length of the target sequence. So if you can see here, like primer 2 begins, and then like primer 1 would be the other boundary. So that's how the primers define the length of the target sequence. So the primers themselves wouldn't so primer 2 does not have an effect on primer 1's Taq polymerase, just as how primer 1 will not have an effect on primer 2's Taq polymerase. So the primers themselves don't really control termination, they control the length just because in the next cycle of replication, when you have this complementary, so you see on the top strand here, it'll have a section complementary to primer 2. So when that strand comes in, it's basically by the third cycle, you will have this, or by the end of the second cycle, you have one side that's the right length, if that makes sense. So that's how the primers define the length. Does that answer your question? Yeah. Right, so restriction enzymes. Yesterday I tried to maybe go through it quickly, but we'll just go back from the start. So, restriction enzymes are biological scissors. And what they are they're endonucleases. So what they do is they break phosphodiester bonds within a nucleotide chain, as opposed to the ends of a chain. So the other time we saw endonucleases was in NER, um, like nucleotide excision repair in, I think, the slide or the lesson before this one. So what do they do? They're found naturally in bacteria, and they're essentially the immune system of bacteria. So they protect the bacteria against intruding DNA from other organisms. So phages, which are a type of virus or other bacteria, so they can recognize short nucleotide sequences in the foreign DNA and then cut the phosphodiester bonds of both strands of this foreign DNA, basically rendering it harmless. Okay, so restriction enzymes have to cut at what we call a restriction site. And each restriction enzyme has a specific sequence that it recognizes and a specific location on this sequence that it cuts. The restriction site is a sequence that is recognized and cut by the restriction enzyme, and it has a couple characteristics. So it's normally 4 to 8 base pairs in length, and it's palindromic. So the same sequence on the complementary strand. So if you have a sequence on the template, the same sequence is found on the complementary strand in opposite orientation. So if you look here, GAA-TTC is GAA-TTC going the other way. So the actual cutting that a restriction enzyme does, we call the process of the cutting digestion. So the enzyme cuts at the same site on both strands. And it creates something called restriction fragments. And these are pieces of DNA that are, created by restriction enzymes cutting the DNA. What it means when the enzyme cuts at the same site on both strands is that, let's go back here. If we have a restriction enzyme that's going to cut this strand, change the pencil, and it's going to cut it, let's say, between these two A's. So if it cuts between these two A's on, we'll call this the top strand, then on the bottom strand, it won't cut here. That doesn't mean same site. By same site, it means it's going to cut here again. So in terms of the sequence GAA-TTC, it's going to cut between the two A's on both strands. And this example applies to if it's between G and A, then it'll be between G and A. If it's between A and T here, then it'll go between A and T on the other side too. So what does this mean? Or why are we explaining this? Because basically the resulting fragments can have what we call sticky ends or blunt ends. Sticky ends are a single stranded end of the restriction fragment, and blunt ends are straight ends without any single stranded regions. So let's take a look at some examples. So in this example, a restriction enzyme is going to create 5' overhang, or sticky end with a 5' overhang. So let's look at this sequence and say that the restriction enzyme cuts between G and A at this restriction site. So what are you going to have? You're going to end up with two fragments that look like this. We have sticky ends because these 4 nucleotides are single-stranded, and it's called a 5' overhang because the side that's single-stranded and basically the sticky side ends in a 5' end. Let's look at another example, a sticky end with a 3' overhang. So in this restriction site, the restriction enzyme cuts here and here, and let's look at the resulting fragments. These 4 nucleotides are once again single-stranded, so this is a sticky end. And you have a 3' overhang because the single-stranded side that it's not, the sticky single-stranded side is ending in a 3' end. So that's why we call it sticky end with 3' overhang, because a 3' end is overhanging. And let's look at blunt ends. And blunt ends is essentially if the restriction enzyme cuts in the middle of the restriction site, then there is no single-stranded area. There is no overhang, so it's just called a blunt end. So these are different examples of restriction enzymes. And there's a specific syntax to naming these restriction enzymes, and they're named according to the organism from which it was identified. So for example, EcoR1 Escherichia coli. So this is Eco is from the name of the original organism, and then R is from strain RY13, and it's the first enzyme in the strain, so we call it EcoR1. Here we have BAM-H5, not 3. Right. Any questions about restriction enzymes? Cool. So restriction enzymes will come back later when you guys learn about something called gene cloning. We use restriction enzymes as a very powerful tool to manipulate DNA. So now we'll get into CRISPR-Cas9. CRISPR-Cas9 can get a little bit complicated the first time you look through it, so if you have questions during the explanation, yeah, ask, because I remember when I first went through this, it was pretty confusing and it took a while to really digest all the information here. So what is CRISPR-Cas9? I'm sure a lot of you guys may have heard about this very important technology. Actually, I think CRISPR-Cas9 is outdated even at this point because it's so it was so bleeding edge when it was first discovered. Anyway, it's really cool. So what is it? It's a ribonucleoprotein complex of CRISPR RNA and the Cas9 endonuclease. What it does is very bare bones explanation is CRISPR RNA targets Cas9 to a specific complementary region on a foreign DNA, and then Cas9 will make a double-stranded break at that location. This is the basic overview of what the CRISPR-Cas9 system does, but the effects or the uses, it can get a lot more specific. So this system exists naturally as the bacteria's immune system to destroy phages. And there's a biotechnology application to CRISPR-Cas9 because using CRISPR-Cas9, you can manipulate the RNA sequence to target whatever you want. So the CRISPR RNA sequence, you can basically manipulate it to target any DNA that you could want. So I don't know if you guys have learned what loci or locuses are, but it just refers to a specific location on the DNA. That's what locus means. It's a location, pretty much. And CRISPR is actually an acronym. So CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. So it's really long, but basically what it means are they're repeats on the DNA. At this CRISPR locus, they are repeats of DNA where they are regularly spaced, they're identical, and they're palindromic. So if you had the host genome and this specific area we're looking at, the CRISPR locus is here, and these clustered regularly interspaced palindromic repeats are these black things. So, spacers are not the repeats, but spacers are also really important. And the spacers in this diagram are the colored boxes, and they're basically DNA memories of previous infection by viruses. So, there is a mechanism to how CRISPR-Cas9 works. So it's basically separated into two large steps. Step 1, immunization. Step 2 is the adaptive or acquired immune response. I guess A and B works better because there's numbered between A and B too. All right. A1, acquiring foreign DNA. So the first step to immunization, so A is all going to be immunization, is acquiring foreign DNA. So just by chance, bacteria would get infected by viruses. And sometimes the bacteria actually survives the infection. And then what it does is it takes the viral DNA and places it within the CRISPR locus as a spacer. This action is performed by the Cas complex. So they're proteins from the Cas genes that we saw earlier. So over here, the Cas genes. So these Cas genes code for polypeptides and proteins that will create something called the Cas complex. And the Cas gene are CRISPR associated genes over here. So when new viral DNA is implanted into the CRISPR locus, a new black box or a new repeat will follow the spacer, always. Step 2 of immunization. You transcribe to RNA the CRISPR, or basically the CRISPR locus. The entire CRISPR locus is transcribed to RNA. So, yeah. When the CRISPR locus is transcribed into RNA, we call it pre-cRNA or pre-CRISPR RNA. The palindromic nature of these repeats forms hairpin loops. So I think remember when we looked at was it transcription where hairpin loops are formed? Palindromic nature of sequences form these hairpin loops because of the attraction between the nucleotides on basically two sections of the RNA here. And these hairpin loops are pretty important because these hairpin loops are basically like physical separation between these spacers. Because these spacers are not identical, but these repeats are. Alright, step 3. After we have this pre-CRISPR RNA, we process it, or it's processed and cut into pieces to form actual CRISPR RNA. Each CRISPR RNA contains one spacer And this processing or this cutting action is performed again by the Cas complex. So if you look over here, it's essentially cut at this point. And what you get is CRISPR RNA. So if you look here, this is all called pre-CRISPR RNA, and after being processed, these are all called CRISPR RNA.
I have a quick question.
Yep.
It says each cRNA contains one spacer. Does it also have to contain one hairpin loop?
I think so.
Okay.
But I think the reason why they don't say one hairpin loop exactly is like, if you look here, like we call this the entire thing the hairpin loop, but when you actually cut it, I guess maybe there's like this residual, like it doesn't just like cut here and here. So maybe that's why they focus on the fact that it needs one spacer and not one hairpin loop, because the spacer is what is important here. But yeah, it would contain a hairpin loop each. Okay, so after you have this CRISPR RNA, the CRISPR RNA is loaded or placed into a Cas9 protein. So with this, immunization is complete. So let's look at a quick summary. Bacteria is infected. It survives, and it is then able to place this viral DNA into the CRISPR locus as a spacer. And remember that each spacer has to be followed by a repeat. Next, this entire CRISPR locus is transcribed into pre-CRISPR RNA. And then, oh, I forgot to mention that the integration or the adding of this viral DNA into the CRISPR locus is done by the Cas complex. Okay, and after transcription, you have pre-CRISPR RNA. The processing of pre-CRISPR RNA into CRISPR RNA is done by the Cas complex again. And then after you have these CRISPR RNAs, they're loaded into these Cas9 proteins. Alright, so after immunization, once the CRISPR-Cas9 mechanism is ready to go, then it can go through something called this adaptive or acquired immune response. So, if the same virus that this bacteria has survived tries to infect the cell again, the bacteria has a Cas9 CRISPR RNA complex that is complementary to the viral DNA. So, when this, let's say, this green virus comes and injects its viral DNA again, CRISPR RNA binds to the complementary viral DNA, and then Cas9 makes a double-stranded blunt end break, destroying the viral DNA and basically rendering it harmless. So let's look at the system. Blah blah blah. We know all about immunization. This step, the CRISPR-Cas9 system is ready to go. So the virus, let's say this green virus comes back, and the bacteria is like, no, I'm ready this time. So what happens is this Cas9, CRISPR-Cas9 complex comes here. The CRISPR RNA has a complementary sequence to this viral DNA, and then it comes here, bonds to it, and then Cas9 will make a double-stranded blunt end break, and then the viral DNA is rendered harmless, and the bacteria is okay. Cool.
Wait, what's the point of the hairpin loop then?
So I think the hairpin loops, my best guess would be that the hairpin loops exist to allow the Cas complex to know when to cut. I think that
Okay.
would be my best guess.
Okay.
That's a good question. Maybe ask Miss Law, or I can try to look it up and give you an answer. But that would be my best guess. So, now that we know how CRISPR-Cas9 normally works, now we can talk about the gene editing applications of CRISPR-Cas9. So, instead of using viral RNA with Cas9, if we use an RNA sequence that is complementary to a gene of interest, This will guide Cas9 to the desired region. And then Cas9 can cut the chromosome in a spot that is complementary to this guide RNA that we've created. And then this could make gene editing less expensive and time consuming and more precise. So let's take a look here. This could be a little bit more difficult to understand, but if we look here, step 1, guide RNA is created. So this is a hairpin loop, and this is this guide sequence. So this would be the spacer. And then this guide RNA is added to the Cas9 protein. Step 3, the guide RNA binds to the DNA at the gene of or the sequence of interest. And then Cas9 makes a cut. And then after that, another piece of DNA can be swapped into the place of the old DNA, and then enzymes repair the cuts, and theoretically, you have edited DNA. Does this make sense? Any questions?
I don't understand how new DNA is like swapped in. Is
So
that what you said?
DNA being swapped in is not the job of
Oh,
CRISPR-Cas9. So I'm assuming what they mean is they would have a separate process of adding DNA in.
okay.
Because yes, you're right. DNA being swapped in is not the job of CRISPR-Cas9. CRISPR-Cas9, the advantage here is that this guide RNA can bring the Cas9 protein to a specific spot on the sequence or the gene of interest, and then make a cut there very precisely.
Okay.
But editing the gene or adding more stuff, if you can see on the other diagram here, after the cut, then it's up to scientists to now edit the gene how they wish to.
Okay.
Yeah. So let's look at some CRISPR hacks are basically some applications. It's an easy way to do a knockout study. So it's an easy way to inactivate a gene. And that's pretty simple to understand because if you make cuts here and the DNA, basically this gene is inactivated because it's cut, right? So it can't be transcribed into a protein and then, sorry, transcribed into RNA and then translated into a protein if the gene is cut out. You can also replace this region with other DNA, and you can actually also add fluorescence tags to DNA using CRISPR-Cas9. So yeah, and this is just another example. With all this CRISPR hacking information, it's a little bit harder to understand, but these are just different ways that we manipulate Cas9. I don't think we were ever tested on this, to be honest with you. I don't remember being tested on this. Epigenetics, you guys haven't even learned yet. CRISPR inhibition and activation, I guess that this stuff. Okay, so let's skip over the CRISPR hacking for now because yeah, I think we can move over this as long as you have the idea of how CRISPR-Cas9 is supposed to work. Alright, so now let's talk about gel electrophoresis. So what is gel electrophoresis? Remember David yesterday, you asked the question of after you have amplified your DNA, how do you use a gel to see it, right? Visualization. So gel electrophoresis is how we visualize DNA. So what is it? It's the size separation of nucleic acids, or sometimes even protein, by moving them through a gel medium using electric current. Oh, it can also be used for protein. Yes, added. So how does it work? DNA is negatively charged. We know this because of the phosphate groups on the backbone. And thus, if you run a current across this, basically the idea here is that because DNA is negatively charged, it will move toward a positive electrode. What you're doing here is in this gel, you've added a mixture of DNA molecules of different sizes. And what you're going to do is, using a power source, you're going to run a current across this gel such that the end with DNA is the cathode, and then the other end of this gel is the anode. And when you run a current across this gel, the DNA, which is negatively charged, is going to move towards the positive electrode. And the idea here is also that the DNA molecules here are of different sizes. So because they're of different sizes, they're going to move at different speeds toward the anode. So, the first step to gel electrophoresis is making the actual gel. So liquid solutions of the gel medium, which can be many different chemicals or many different like substances, although there are two common ones that you guys will learn about, is poured into a mold and allowed to set and solidify. There's something called a comb. Wait, do you guys learn about the comb? I guess we'll learn about the comb in a second. And it's in this picture, it's telling you to notice that the comb is placed at one end of the gel. When it is removed, it creates the wells where DNA is loaded, and the comb is this thing. Basically, what it's asking you to notice here is that the combs are basically like, imagine you stuck a comb into a gel, and then the gel was solidified against this comb. And once you take the comb out, it'll create like wells, so little like holes in the gel. And this is where DNA will be loaded, and we'll learn about loading in a second. So, in this picture, you have what we call a horizontal gel, and this is because the DNA is going to move horizontally across the gel. In a vertical gel, DNA will move vertically. So this is a good place to look at the comb. So imagine that you set your gel and had it solidify, and you had a comb in here. And basically the teeth of the comb were going into here. When I talked about wells, these are the wells. So if you had your solution of DNA, they would go into these wells. Let's see how much in detail the next slides go. Okay, let's keep moving on then. Alright, so before we talked about gel mediums, or remember earlier we said that you pour your gel in, but there's actually different types of gel mediums. So we're going to learn about agarose and polyacrylamide. The difference between these two gels is essentially their resolving power. What resolving power is the gel's ability to separate substances of different sizes, or nucleic acids or proteins of different sizes, and the accuracy of the separation. So what I mean is that agarose, which is seaweed extract, has a lower resolution than polyacrylamide. So if you had DNA, let's say, of very, very small increments in base pair sizes. So the way that we would basically say that one DNA fragment is larger than another is the number of base pairs that this DNA is, or how many base pairs long it is. Agarose's lower resolution means that when you have DNA fragments that are closer in length to each other, its ability to show them like separate from each other in this gel electrophoresis is going to be lower than polyacrylamides. Maybe it's best to explain how this gel electrophoresis thing is going to work then. Yeah, I'm sorry, maybe I should have explained that in the first place. So, you look here, this is probably a better example. In this vertical gel, you can see the wells. So they've been drawn out with this marker here. And inside the wells are this blue solution. So in this blue solution, you have your DNA and uh, and a pigment. And this pigment will be of a certain size. We know how quickly this pigment is going to run across the gel because we've tested it before. And basically the way to say it is this, let's say we have like a blue pigment in here, that's like a 500 base pair equivalent. So it will move at the speed that a 500 base pair DNA fragment will move. What we do here is we put these DNA solutions, and in these DNA solutions will be DNA fragments of different sizes. Most of the time we'll know what sizes to expect based on where we got this DNA from, but the pigments here are basically here to know our reference point. So what's going to happen is that the pigments are going to move down, and DNA, which we won't see, is also going to move down. And that's why we talk about resolution. Because in the final product, let's say we had like a 500 base pair fragment and like a 515 base pair fragment, that's a very, very small difference, right? So if we wanted to see that on our gel in the final product, then we would choose to go with polyacrylamide. But if we know we're handling a 500 base pair fragment and like 1000 base pair, then that's like a very large difference. So then we know that we can use agarose. So what is gel electrophoresis doing? It's basically separating nucleic acids or proteins by size. The gel medium is semi-solid, so it provides resistance for DNA movement. The more concentrated a gel is, the higher the resolving power of the gel. So what you can think about it is that it's semi-solid, so in it there is going to be like solid particles of the gel, and between these particles, or there will be like small holes for the DNA to pass through. And that's basically how you can think about the sentence here, providing resistance for DNA movement. Oh, it's literally explained in the next slides. Okay, so separation by size. Short DNA will move through the gel more easily, and thus in the same time as long DNA, if you give the same time to travel, it will travel further. Long DNA will move through the gel slower. Because of its size, it will get caught in the web of polymers that make up the gel, and therefore it will not move as far. So all the DNA is starting at the negative, or the cathode, so it's starting at the negative end, and remember it's moving this way. So basically the bands that are closer to the negative side are the longer molecules, and these bands that are closer to the positive side are the shorter molecules. So let's look at why we learned about restriction enzymes. So let's say we take your genome, or we take a specific gene or location on your genome. We use restriction enzymes to cut your DNA into different fragments, or cleave DNA into smaller segments of various sizes. Then these DNA segments will be loaded into wells in this gel. The gel will float in a buffer solution within a chamber between two electrodes. When an electric current is passed through the chamber, DNA fragments will move towards the positively charged cathode. And as we learned on the previous slide, smaller DNA segments will move faster and further than larger DNA segments. So, why do we have the buffer? The buffer provides a medium for the flow of electric current, and it also prevents the gel from overheating and drying out. And the colored dyes that we mix with DNA that you saw earlier, because DNA is not blue, DNA is clear normally, the dyes are used to track the distance traveled on the gel by DNA. And they also have a job of increasing the density of DNA so that it will sink to the bottom of wells. Because remember that this gel is floating in a buffer solution, so DNA has to be denser than this buffer solution to float to the bottom of the well to move through this gel. And dyes, which we'll call loading dyes, have a very, very important job. Because normally, when you load your DNA into wells, if you do not have loading dyes, you cannot see DNA. DNA will be clear. So if DNA is clear, how do you know when to stop your gel electrophoresis? If you don't have loading dyes, you don't know, right? You can only guess. But guessing is bad. So that's why we have something called loading dyes. You see the purple and the blue? These are two different types of loading dyes. Oh, do they? Okay. So essentially what we're doing is, let's say in this solution of DNA, let's say in a single well, we know that we'll have fragments between 100 and 700 base pairs. So 100 will be the smallest, they'll move the fastest, and 700 will be the slowest, and they'll basically move slower than everybody. So in order to know when to stop our gel electrophoresis, we will have to have a loading dye that will move faster than our shortest DNA fragments. So if this was holding 100 to 700, we will have to make sure that we have a loading dye that will move faster than 100. So let's say that this purple loading dye is going to move at the speed of, I don't know, 80 base pairs, just for sake of example. So now we know that this purple line of loading dye will move faster than any other DNA segment that we will have in this solution. So what do? Now we know to stop the gel electrophoresis when this purple band gets to the bottom. If we didn't, we wouldn't know when the DNA would move off the gel. And if DNA moves off the gel, we lose that information. You've basically ruined your gel. So now, because we have these loading dyes, we can move them. We can stop it when this purple gets to the bottom. But just because we stop it when the purple gets to the bottom doesn't mean that we're done. Because the loading dye doesn't make DNA on the gel visible. DNA will still need a separate stain for us to see it. And this is where we learn about DNA staining. So, ethidium bromide is a compound that is used to stain DNA. It's a chemical that binds to DNA, and it's also carcinogenic, but it glows under UV light. So if you add ethidium bromide to stain your DNA, and then you use a UV light box, then you can see the fluorescent DNA bands. So let's take a look at this. This is DNA stained with ethidium bromide, exposed under UV light. So you need to stain DNA to see these separate bands, because before you stain DNA, all you see are your loading dyes. Does that make sense? Any questions? Yeah, so I think next lesson we'll learn about DNA fingerprinting, which is an application of gel electrophoresis. Oh, what are the nerdy gifts? They're just different gels that have been run. So let's say that this was your DNA at like a certain locus, and then it was ran on a gel and then stained and taken a picture of, and then like somebody sent it to you. I think these are cookies. So I mean that's why it's a nerdy gift. These are just different gel stainings. Yeah. So next
So,
lesson, yeah, sorry, David, go ahead.
just a question. Once the loading dye reached the bottom, what you're measuring is how far the DNA got, and that's going to tell you how many base pairs are in the DNA?
Essentially, what we also do is, I forgot to mention that, we have this. So you see how these are green? Everything else
Yes.
is red. So
Yes.
your greens are basically your control. So if in 1, 2, 4, 5, 6, 8, 9, 10 you don't know the fragments that are contained inside, in your 3 and 7 you will basically have fragments of all the different sizes, and once you stain your DNA you will compare the base pair links to the columns 3 and 7, or to the wells 3 and 7.
All right.
I think it might become a lot clearer uh, maybe in this example. So let's say that in this example, in this gel electrophoresis, the solution in this well is your control.
Okay.
And you know that like this would be 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, 120, 130, 140, 150. And because you put it in that way, then with this well, once you're done your staining, you can compare the lengths of all these other bands just by looking at where they are relative to the bands on this column.
Oh, okay, that makes sense.
Yeah. So you can't really know an exact number, but you just know using this well that you put in yourself. Yeah. Good question. Yeah, you guys would do this at the science center, but unfortunately, I'm pretty sure that trip was canceled.
Oh man, that looks so cool though.
Yeah, it is. You don't actually do staining yourself, but you do get to see your own. You get to run gel electrophoresis with your own DNA, so it's pretty cool. All right, any other questions? I'm seeing Nathan typing, that's why I'm waiting.
There's so much pressure.
No, it's not pressure. Just take your time. Okay, he's fine. All right. All right, that's it, guys.